Here at the back. Yes, good. Well, welcome to all those with and without jet lag. I feel like I had jet lag, but only due to the Radisson Blue at Stansted Airport. That gave me a room with 3 beds, one on wheels that I kept hitting on the way to the toilet, etc. But I managed to get here cheaper, in actual fact, than travelling from Paddington to Oxford. The ticket from Stansted Airport through Paddington to Oxford was a third of the price, just in case you think you might stay in London and then travel on. Anyhow, enough of that. Welcome to Organelle Pharmacology on behalf of the BPS, the British Pharmacological Society, emphasising this just in case there's any biochemists in the room, or even physiologists. The British Pharmacological Society, of course, supports a great deal of science, and to the ECRs, and this meeting is primarily targeting ECRs, we decided that regional meetings, visiting the great universities of this country, died away. And that was a loss to early career researchers, because unlike me, I was thrilled the first time I visited Cambridge University. I can remember then Oxford. Birmingham was even a thrill to me at the time. And all of a sudden we went to big meetings, and many people missed out on that opportunity. Staying in halls with a communal bathroom. Fantastic. So, we've brought this back, and with the intention of highlighting the society as well, and its work. And I want to highlight its work because I never really got involved with the BPS on an administrative level. I attended the meetings, I thought it was all great fun, had plenty of beer. And then one night, just to give you some background, my wife had been given 2 Dover sole, and they hadn't been filleted or even descaled. And the guy, I think he was having a laugh, the fishmonger said, "Oh, descaling is easy, you just get a knife and do that." Well, dinner was delayed, and my wife eventually came in and told me dinner was available with scales in her hair, and they were all over the kitchen. Took her hours to clear up, but I wrote. The BPS had sent me an email, and I give you this background because I think it's important that I didn't get involved before. And I was in a bit of a grumpy mood that weekend, so I just put down everything I thought, because they said apply to do this, that or the other. And I thought I'd go for VP for meetings. And I put everything down that I didn't like about what was going on at the moment. I never thought for 1 minute anybody would vote for me. But they did, and that was a shock of my life, because all of a sudden I had to do the job. And I came in, and I met all these people, some of them I'd seen before, and I've realised over the last 3 years the workings of the society, and Isabel and the team there, who produced the main meeting and the regional meetings, and it's absolutely fantastic. It's been a thrill. Thoroughly enjoyed the last 3 years, and I'm going to miss it terribly. And I encourage, they're not ageist, you don't have to have grey hair like me. You can apply at the age of 30. You can hold meetings like this. Early career researchers, I'm speaking to you. It doesn't have to be the head of department like Anthony kindly agreed to, and Fran, host this meeting. So, do get involved, become a member, and don't think for 1 minute that you can't do the jobs that are advertised. And they are good fun, so I encourage you to get involved. There's plenty of support for members. We've got discounted rates, of course. Bursaries, travel bursaries, not just for the UK, but to go abroad. For example, the World Congress of Pharmacology in Melbourne, Australia this year, which I'm going to, and I hope many of you are. And we have several meetings each year, finishing always with the meeting of the year, this year, of course, Pharmacology 2026 in Manchester. And we've managed, with the work of Isabel and her team, we've got, since lockdown, the attendance has gone up from 450 to 890 last year. And we're going to break 1,000 this year in Manchester. So please come along. Very vibrant, full meeting. And it's been really good fun. And with that, I finish with our journals, which I'm sure you all know. And I encourage everybody that's here that thinks they've got a small paper or a review article Anthony can be the lead editor. I'm happy to assist. We have Charles Wally, head of journals, out at the desk. And a special issue from this meeting would be greatly appreciated because it is an important area. And that can be reviews and small articles that are worthy of rapid publication. With that, I hand you to your host and mine, the Don Gallio.
Thank you very much, Mark, and welcome all of you to Oxford. It's great to see so many of you here, and I hope you'll have a really enjoyable 2 days. We've got a sort of action-packed day-and-a-half meeting. We've got some wonderful, stellar speakers, and I hope you'll all have a really edifying and great time here. So the topic of today's symposium is Organelle Pharmacology, a new frontier for drug discovery. This is an area that we're particularly interested in this department, and we are in the process of setting up, under the leadership of Fran Platt, a centre for organelle drug discovery in part of this building. So just a word about organelles. Eukaryotic cells studied with a number of different structures which we call organelles, and they all have different functions. They compartmentalise the different functions of a cell. So, for example, we're going to hear today about mitochondria, which is the powerhouse, energy powerhouse of the cell. We're going to hear quite a lot about the endolysosomal system, which traditionally was thought as a trash can of the cell, but it has many other functions, particularly in cell signaling. And of course, this lattice work of the endoplasmic reticulum, which has a variety of functions, not only calcium storage, but its role in protein trafficking and so on. But, you know, it may not come as a surprise that these organelles do not work alone in isolation. No organelle is an island. There are ways in which they communicate, and this communication is extremely important. It can occur via signals or actually at membrane contact sites, and this is a theme we're going to hear about during this meeting. Now, when individual organelles go awry, disease ensues, and sometimes a defect in one organelle will have a knock-on effect on other organelles. And this is something which we're going to think about over the next day and a half. So from the website, what we're going to do today is think about the organelle disease connection, and we've got a theme on that. We're going to talk about emerging techniques for studying organelles and how drugs may modulate their function, and also novel therapeutic strategies. And I think you've all got the programme online, so we've got 3 themes over the day and a half. We're going to start off with organelles and disease, chaired by Margarita Ruiz this morning. And we have invited speakers, and we also have shorter oral communications. And then we have lunch and poster viewing back in the seminar room where you've all had coffee. And this afternoon's session starts again at 2 o'clock, and the theme is drugging organelle targets. And we're delighted to have Martin Gunthorpe or Tiffany to chair that session. Again, same pattern. We've got invited speakers and some shorter oral communications. After today, after the closing remarks, there'll be a drinks reception up in the common room of this department where drinks and I think some edibles will be served. So tomorrow we start, I'm afraid, at 9:10. And the final theme is emerging techniques for studying drug effects on organelles. And we're delighted to have Sandip Patel, who's going to chair that session. Again, invited speakers and short oral communications. And then we conclude with closing remarks and then prizes for the best, I think, communication and posters. Is that right? Good. So the BPS just asked me to mention what we should get from this day and a half, our learning outcomes. So we hopefully will better understand the roles of cell organelles in health and disease. We will understand the current state of the art, identifying and studying organelle drug targets. We'll appreciate the challenges of targeting intracellular sites. And hopefully we will be mindful of being able to translate our new understanding to the development of novel therapeutics. But my final remark is you are here in the Department of Pharmacology in the University of Oxford, which goes back more than a century. But this is a new building which was opened by one of our former students, Sir John Vane, who was a Nobel Prize winner in 1982, and he got this for working out the mechanism of action of aspirin. And he was rather ahead of his time, because aspirin targets organelles. It targets proteins, enzymes, which are actually in the endoplasmic reticulum. And thinking about aspirin explains some of the challenges that we're going to be thinking about today. The drug has to enter the cell, and then it's got to specifically target a particular process or protein on an organelle. So my final comment is grateful thanks to our sponsors today, BSIS, Aurea Bioscience, Sofian, or Tiffany Therapeutics. A very warm thank you to you for sponsoring the meeting, because without you, the BPS, we couldn't put this on. And of course, I should thank Bonnie and the team at the BPS and Mark for his kind words of introduction, for facilitating this meeting. And I'd like to thank Fran and all our colleagues in the Department of Pharmacology for hosting this event. So I'd just like to say I hope you have a really productive, enjoyable and collaborative time over the next day and a half. So with that, I'll hand over to our first chair, Dr. Guida Ruiz, who's group leader in organelle biology at the Alzheimer's Drug Discovery Centre up in the University of Oxford Medical Science Division.
Good morning, everyone. It's a real pleasure to welcome you all to this first session. I think this first session really sets the tone for the rest of the program. So we'll hear speakers highlighting the fundamental biology of underpinning organelle function, and now we are translating that knowledge into the development of new therapeutic interventions. So, and we have a fantastic lineup of speakers that are experts in this field. So I want to keep this brief, but before I start, I just want to have a note for the speakers. I will stand up 3 minutes before the end of your allotted time, leaving roughly 5 minutes for questions so that the audience can ask questions at the end of each presentation. And if we run out of time, I'm sure there will be plenty of opportunities to interact during our refreshment breaks. So, without any more delay, I will be welcoming the first speaker. So the first speaker is Professor Fran Platt. Fran is really a leading expert in lysosomal storage disorders, specifically in glyphosphingolipid disorders. And her work has contributed to the development of one of the first approved treatments for these conditions. Fran is a fellow of the Royal Society and also the Academy of Medical Sciences, and she currently serves as the head of this department, the Department of Pharmacology here in Oxford. So today she will be speaking on the lysosomal diseases from molecular pathways to therapeutic impact. And I just want to welcome you from, and I'm really looking forward to your presentation.
Great. Well, good morning, everybody. Thank you very much for that kind introduction. I'm told that if I press the advance button, my slides will appear. Possibly not. Oh, okay, great. So I'm going to split this talk into two sections that are interrelated. And it's really to illustrate the fact that if you take a deep dive into disease processes, you learn a lot about basic science. And in some cases, you can use that knowledge to innovate treatments. But also, if you do innovate a treatment, when you start using those treatments, that also sheds more questions about the basic science of the disease process. So it's very much a sort of an organic process, and it's taken about, 30 plus years of my career to do this, but I'm just going to share some current things that we're interested in and the state of the art with the therapeutic side of things. So I'm going to focus entirely on one disease to keep this simple. And I'll begin by disclosures. I'm an academic co-founder with Anthony Gaglione and Grant Churchill of a company called Intrabio that we set up because we were convinced that a drug would work, but we couldn't persuade anybody to invest in it. So we ended up setting up a company to do that. I worked closely with Orphazyme on one of the drugs that I'll be talking about, which is a Danish biotech company, and that was work that was funded by them and also by the Wellcome Trust. So, Niemann-Pick C is a disease that we've been obsessed with for many years. It's endlessly fascinating, and I'm going to hopefully explain why that is a disease that we've become really interested in. So, it's a rare autosomal recessive lysosomal disease, so nothing particularly unusual there. It can be caused by mutations in two independent genes, NPC1, which is most cases, and NPC2. And there are multiple lipids stored, including glycosphingolipids and other sphingolipids that we're interested in. And at the cellular level, there's a block in late endosome-lysosome fusion and trafficking problems. Now, if you look in the lysosome at what these two proteins that are encoded by the two genes are, they're very, very different. NPC2 is a soluble cholesterol binding, probably other lipid binding, mannose-6-phosphorylated archetypal lysosomal protein. Whereas the NPC1 protein is this large multi-membrane pass protein that is one of the few surviving orthologs of an ancient family of bacterial transporters called R&D permeases. And what those permeases do is they move substrates from inner membranes to the outer membrane, and then this family of proteins act cooperatively in bacteria with another protein which gets substrates across the periplasmic space. That other protein does not exist in mammalian systems. So we've been interested in what the role is of this conserved role of these two proteins that we think are acting cooperatively. And when we inactivate NPC1, we get a very strong sphingolipid signature. Sphingosine is the first storage lipid that appears within a few minutes, and so we've postulated that NPC1 in part is a sphingosine transporter. And obviously sphingosine, when it leaves the lysosome, can become phosphorylated, which is a key signalling molecule. But what's really relevant to today's talk is that this sphingosine storage reduces levels of calcium in the lysosome, and this is because of a filling problem. So there's a defect in filling of the acidic store with calcium, and it's this that's responsible for the block in late endosome-lysosome fusion, because the lysosome is not releasing as much calcium as it should. And then you end up with cholesterol and sphingolipid storage sort of downstream of that block in trafficking. Now, the sphingosine interaction with NPC1 has been proven by Doris Hoglinger using a trifunctional probe, and so we know that at least sphingosine is a direct binding partner of NPC1 in terms of lipids. Now, the fact that these R&D permeases in bacteria work cooperatively with another protein, they do not work on their own, led us to hypothesize that perhaps in mammalian systems, the NPC1 protein, this multi-membrane pass protein, was also working cooperatively with other proteins. And we worked with Maya Scholdinger when Ali Calesso was in the lab and Maya's at the Weitzman. And she ran 2 biochemical interactome screens in yeast. One was to look at the global picture of what could the yeast ortholog of NPC1 bind. It's the genes called NCR1 in yeast. And we also did one that was limited to the vacuolar membrane to see if the protein was existing in a complex. And to cut a very long story short, with Doris Hoglinger when she was in the lab and in collaboration with Emily Eden, who's here today, we discovered that there was an interacting partner in the ER. And it's a protein called GRAMD1B. It's a bona fide lipid transfer protein, and it interacts with NPC1. And so actually NPC1 in mammalian cells is acting as part of a tethering complex, which bridges the lysosome to the ER. And it's this was really resonated with us because the problem with lipid transport in NPC is from the lysosome specifically to the ER. So this was the first indication that this is a contact site-based disease. In Emily's beautiful electron microscopy that she did in that paper, this is showing the lysosome in contact with the ER. The NPC1 protein is preferentially at these contact sites, shown here. But what Emily also noticed, and this will be relevant to the very end of my talk, is that she noticed that when you don't make proper contact sites in NPC between the lysosome and the ER, you make excessive and really fascinating looking contact sites between the lysosome that's here and mitochondria. And this then means that lipids from the lysosome end up in mitochondria, and we know that there's secondary dysfunction of mitochondria in Niemann-Pick C disease. What I want to focus on is the other screen today, which was the screen that we did to look at the interaction of the NCR1 protein with other vacuolar proteins. The yeast vacuole is the equivalent of the lysosome, and I don't know whether I'm probably standing in the way of everybody sitting on this side of the room, but there was a very strong hit in this interactome screen between NCR1 and a gene in yeast called PMC1. And this got our attention, and Ali, while she was in the lab, showed that you could co-immunoprecipitate NPC1 in the rat brain with one of the isoforms of PMCs in mammalian systems, which is this protein called PMCA2. So we knew that they interacted in yeast, and we knew that they interacted in the mammalian brain. But we now had a problem, because if you look up what PMCA stands for, these are plasma membrane calcium ATPases. And even though the gene in yeast is a vacuolar protein, it has that name because it's come from the mammalian side of things. And that cell surface isoform of these PMCAs is responsible for extruding calcium from the cytosol into the extracellular fluid. And as I say, it looks as though it probably evolved as a lysosomal protein. And we wondered whether or not there was any possibility that mammalian PMCAs had another role, not just at the cell surface. Is it possible that they also have a lysosomal function that had not been appreciated before? And Maria Fernandez Suarez, who's a fantastic senior postdoc in the group, looked at the PMCA sequence, and she became fascinated by the fact that this protein can be differentially spliced. And there are many, many isoforms that can be generated, particularly at this splice site C. And to cut a long story short, what she found is that, and Andrea Bellavio will like this, there's a clear sequence, a TFEB binding site in this PMCA2 gene, which is called ATP2B2. So that's the gene that encodes PMCA2. And also there's a lysosomal targeting motif. So what this was suggesting, that there are splice variants of this protein that function in a completely different compartment from the canonical cell surface form. But having said that, we think that the lysosomal form actually evolved first, and with work with Chris Ponting and colleagues, we've shown that that is likely the case. Now, in mammals, there are 4 forms of the PMCAs, 1, 2, 3, and 4. 1 and 2 have got both the clear sequence and the lysosomal targeting motif. PMCA3, which is another brain isoform, has just the lysosomal targeting motif. And 4 is a sort of ubiquitous, probably the cell surface isoform. When Maria looked at the expression of PMCA2 and these are in PC12 cells differentiated into neurons, The green is PMCA2, red is NPC, and you can see that there is no cell surface staining, but there is coincident staining. So we were really, really fascinated by this because this suggested that this could explain why there is a calcium defect in NPC for the first time. And that's because when you model PMCA2 in complex with, this is the NPC1 protein here, this is the limiting membrane of the lysosome, and you can see this very nice complex that's formed with PMCA2 and NPC1, and the direction of travel of calcium would be from the cytosol into the lysosomal lumen. And this is just showing you that in yeast, when Ruben Bush, who's a student in the lab working with Simon Newstead, they've modelled the PMCA, the PMC1, the vacuolar version, and again, you can see a lot of similarity in these structures. Maria then worked with Samdip Patel to show that if you knock down the ATP2B2 gene, you have much less calcium released from the lysosome compared to the control wild-type situation. So what these experiments have shown is that there is enormous complexity to this NPC pathway. It's involved in contact site formation. It forms a complex that orchestrates lipid efflux and calcium influx, and we think that's probably happening at ER lysosome contact sites. And in NPC, you don't make those contact sites, so you're not filling the store properly. So I'm going to leave the basic science part there and switch gears to how the complexity of this pathway has led to a really unusual situation in terms of therapies for this rare disease. We've actually got multiple approved therapies, and what's most appropriate for this discussion today is that they're all small molecules. And all of those molecules are potentially applicable to more than one disease. So in the lysosomal disease field in general, many of the diseases are caused by deficiencies in lysosomal enzymes. And so some of the first new era of biologic drugs, some of the big blockbuster billion-dollar-a-year Genzyme products of old were recombinant enzyme replacement therapies to treat things like Gaucher and other lysosomal storage diseases. The problem with those therapies is they don't cross the blood-brain barrier, and also they are totally disease-specific. So you can only fix one disease with one product. And we were very interested in the possibility of getting a new generation of small molecule therapies that would go everywhere, hopefully within the body. And I'm just going to give you 3 examples today in the Niemann-Pick C space to just illustrate some of these points. So if you have a complex pathogenic cascade, it can be quite overwhelming in the cellular complexity, but it gives you a wealth of opportunities for intervention. And the one thing that I've learned is you don't have to fix the whole problem to have a therapy that's beneficial. Even if you can modify one aspect of the disease, it can be a game changer for patients living with these disorders. So we are in a situation where the first drug was approved in 2009 for Niemann-Pick C, and we've had 2 new approvals in 2024, actually in the same week by the FDA for 2 new small molecule therapies. And so I'm just going to review with you where we are and then talk about one of those in detail. So this is the only disease I know of in the lysosomal disease space that has more than one small molecule. Most diseases do not have a small molecule therapy at all in this space, but NPC is an exception. The first one that was approved is miglustat, which is an amino sugar drug that I worked on years ago as an antiviral. We discovered it's a substrate reduction therapy. It can block the biosynthesis of glycolipids. So if they're building up in the lysosome, a bit like a statin, you can down-regulate this pathway. Oral drug gets into the brain, and it was EMA approved for Niemann-Pick C in 2009. It had previously been approved by the EMA and FDA as a treatment for Gaucher disease, which is a glucocerebrosidase deficiency. The next drug that we worked on was with Orphazyme in Denmark, which was arimoclomol. This drug is approved and called Miplifa. And this is a HSP70 targeting drug that amplifies the HSP70 response. And the exact mechanisms of this remain sort of incompletely understood, but again, it's an oral drug and it was approved quite recently. And then the third drug, which is the one that Anthony and Grant and I have been working on for the last sort of 10 years or so, is acetyl-L-leucine, or ACNURSA. This is a modified amino acid. It's another oral drug, and it's been approved by the FDA in 2024 for Niemann-Pick C, and more recently this year by the EMA for treating neurological manifestations of NPC. So let's just deal with miglustat first. So miglustat is an organelle targeting drug. This is a drug that is a small amino sugar glucose mimetic with an alkyl chain. It has multiple enzyme targets in the cell, but what's really interesting is that most of those targets are not available because they're inside organelles that very tightly regulate the environment, like ER glucosidases, for example. However, based on KI, you would never have picked this as a target, but it just shows that you need to look at whole cells, because in fact the target is an enzyme that sits on an early Golgi compartment. It's this glucosylceramide synthase enzyme, and it has its active site in the cytosol, so it's very, very drug accessible. Whereas the better targets, from a pharmacological point of view, are actually intractable and inaccessible, and actually in reality are not really engaged by the drug. So we developed this drug as a way of down-regulating glycolipid biosynthesis. It's CNS penetrant, and it was approved first for Gaucher and then for Niemann-Pick C. Now, after 10 years of patent coverage and orphan designation, there was a very nice study by Mark Patterson that was published in 2020 that showed the natural history of Niemann-Pick C before the introduction of this drug, and that's the blue line, where the probability of survival of patients, the mean probability of survival, was about 11 years. And that has now been moved out to about 22 years just by this one small molecule. And so what we were then interested in is, can we add other small molecules in that will target other aspects of this pathogenic cascade that will then work cooperatively or synergistically with this drug that's now standard of care. So the next drug that was approved was Miclefa, or arimoclomol. This is an HSP70 amplifying drug, and in preclinical studies in mouse models in our lab, working with Orphazyme, funded by Wellcome, we discovered that in Niemann-Pick C there is a hypomyelination. You don't myelinate the nervous system properly, and this is because of an oligodendrocyte precursor maturation problem. And I think this slide has gone slightly awry, but let me just tell you the bottom line, that if you treat the mice with either a recombinant HSP70 or a drug that induces HSP70 expression, you can see that the green myelin basic protein in the cerebellum is much, much better in the treated animals. This was published as a study in Science Translational Medicine. And then we had a very interesting follow-up paper by James Gray in my lab, who worked on this with Thomas Kierkegaard and colleagues at Orphazyme. And what James discovered was that what we seemed to be doing was increasing a fin kinase-mediated signalling pathway, boosting oligodendrocyte maturation and differentiation. And this is what was rescuing this phenotype. Because when he used a drug, saracatinib, that's a fin kinase inhibitor, it abrogated the effect of the HSP70-based therapy. So this is working in a totally different way, and it's improving the phenotype of the mice, but in a complementary way to miglustat. Now, in the last few minutes, I want to talk about this very unlikely drug, acetyl-L-leucine. How can a modified amino acid be a drug? This has a history that since 1950s in France, this drug is a racemic mixture of D and L enantiomers, has been on the market as you can walk into any French pharmacy and buy this as Tangenil, I think would be the pronunciation, Tangenil. It has very few side effects, and it was used for treating acute vertigo. And it was found by Michael Strupp and Tatiana Bremova, clinical colleagues in Germany and now in Switzerland, that this drug also improved cerebellar dysfunction in a variety of cerebellar ataxias. And Tatiana is a Niemann-Pick C expert, and she included some NPC patients in the cohort that was studied. And they really improved, and they improved rapidly. And so we started working with Tatiana and Michael to try and understand which enantiomer was responsible for this. And we trialled this drug at the same dose that was being used in the clinic. So this is sort of back translation, if you like. And what we were looking for was whether or not we could see similar clinical benefits in the mouse so that we could study mechanism. And to cut a long story short, we found that it was the L enantiomer that was functional, and the L enantiomer was not only improving symptoms, but it was also delaying disease progression, a bit like miglustat does. And Grant Churchill, our colleague, in a really nice study, showed that the acetylation of leucine turns it into a prodrug. And leucine, the natural amino acid, is scavenged by the L-amino acid transporter, which is a fairly specific but very low capacity transport system. By acetylating this L-amino acid, you actually convert it from a substrate for the L-amino acid transporter to a modified drug that can now go up into cells through the monocarboxylate transporter, which is a much more promiscuous family of 14 proteins. They're everywhere. They're all over the blood-brain barrier. They're much, much higher capacity. And so what we're doing is we're flooding the cytosol with the acetyleucine. It's the acyl group is removed. It generates leucine at high levels, which then, we're still trying to understand the details of this, is able to restore aspects of mitochondrial and lysosomal function. Now, to test this hypothesis, we have gavaged animals with C13 labelled acetyleucine or just the C13 non-acetylated version. And then we've measured the C13 leucine that arises after the deacetylation. So we fed with the two independent analogues, and then we're measuring the C13 leucine. And you can see, I'm sure from the back of the room, that in blue at 30 minutes, if you've administered the C13 acetyleucine, you're achieving really high levels at the 30-minute time point compared to the leucine-treated animals. And we think that this is part of the mechanism of action, these supraphysiological levels of leucine that are generated. In the mouse, the effect of this treatment was really interesting because there is altered glucose metabolism in the brain of Niemann-Pick C, but also of a related lysosomal disease called Santoff disease, one of the GM2 gangliosidosis. And in terms of the balance between anaerobic and aerobic respiration, there is a geometric opposite dysregulation in these two diseases. But what was remarkable is that the acetyleucine rebalanced both of those. So this is a metabolic rebalancing drug. On the basis of these studies and the clinical proof of principle using the racemic mixture to begin with, acetyl L-leucine was then trialled in 2 phase 2b trials in Niemann-Pick C and GM2, and it showed a rapid improvement in cerebellar ataxia. I'm almost at the end, Gida. And this then led to a phase 3 pivotal trial in Niemann-Pick C, and this was published in the New England Journal a couple of years ago, and it's a double-blind crossover trial. And the nice thing about this is that each patient then acts as their own control as they go from test to placebo or vice versa. And this is just showing you that when they're on the active substance, they improve in terms of their SARA scores. This is measuring cerebellar ataxia, whereas they deteriorate when they're on the placebo. And this is just showing you the switched arm of that study. This protocol was approved by the FDA, and it led to, with this publication, the approval of this drug by the FDA in 2004. Now, if you remember back to the contact site problem in Niemann-Pick C that we've worked on with Emily over the last few years, Emily's lab have just published a really beautiful paper on bioRxiv that shows, to our amazement, that when you treat cells from patients with Niemann-Pick C with the acetyl L-leucine, is that, if you remember, I said that there's a problem with normal formation of lysosome ER contact sites, and you make excessive lysosome mitochondrial contact sites. Emily's lab have shown very beautifully that these imbalances in contact sites are restored by this drug. And we're trying to understand, or Emily's trying to understand at the moment, the precise mechanism of this. So this is just showing that with the knowledge of the underlying cell biology, you can now look at these treatments and try and understand exactly how they're working. And I think with acetyleucine, it's going to be quite pleiotropic. My final thought on this, and I've just got one more slide, is that when you think about neurodegenerative diseases that we tend to all study, somebody studies Alzheimer's, somebody studies Parkinson's, we're in the rare space. when you actually come down to it, there's a huge amount of overlap and convergence. Every neurodegenerative disease has undegraded waste of some sort, whether they're protein aggregates or, in our case, lipids that are building up. They have activation of innate immune inflammation, glucose metabolism in the brain is perturbed, cytoskeletal defects, and altered organelle function. And what we hypothesised was that the acetyleucine, because of the way that it's working in this multifactorial way, is that it may actually be a modulator of not just rare lysosomal storage diseases, but other more common disorders, or things that are not lysosomal diseases but have another etiology. And I just want to share with you that in January of this year, there was an identical clinical trial run to the one that I've just described for Niemann-Pick C, but this time it was in ataxia telangiectasia, which is actually a DNA repair enzyme deficiency. It's not a lysosomal disorder. But there is a lysosome mitochondrial axis that is disrupted secondary to that in this disease. And there was benefit reported in this clinical trial. And so Intrabio, the company that I was discussing at the beginning, have now filed this with the FDA, and we're hoping that if all goes well, this will be approved for ataxia telangiectasia in October of this year. And this is a disease with no approved therapy at all at the moment. And also, unlike Niemann-Pick C, most of these AT patients are cognitively well, but they've got a huge physical burden. And so we really hope that this does get approved, because I think it could be a really a really good starting therapy for the ataxia field. So with that whistle-stop tour, I hope it's given you a flavour for the basic science of these diseases, gives you a lot of insight into basic fundamental processes, and also a lot of potential ways in which we can target for therapy. And I'm very happy to answer any questions. And just thank everybody involved, who's a cast of thousands. Thank you.
Thank you so much, Fran. That was a fantastic way to start the day. Frank, can you take questions?
Very, very interesting talk. Concerning the three drugs, I find it puzzling and maybe also scaring the fact that with the exception of miglustat, the other two drugs, it looks like they have little to do, except perhaps for the last slide that you showed, with the mechanism of the disease, specific mechanism of the disease, which is the transport of cholesterol in and out the lysosome. So, meaning that these two drugs would have never been identified. You said it yourself. You said the unlikely drug for. So they would have never been identified with a clever strategy. What do you think about it?
Yeah, well, first of all, just in terms of the fundamental science, I'm probably in the minority on the planet who do not believe that NPC1 is about cholesterol efflux. The cholesterol is moving out at these contact sites, so it's really the contact sites that matter. So the fact that the contact sites are being corrected by acetyleucine is really fascinating, because although this does not appear to be a primary... So in other words, the way I would look at these pathogenic pathways is that you've got your primary problem and you've got all of these secondary collateral problems down the street. And you could argue that you've got to be as close to the top of that pinnacle as you can to have maximal benefit. But we thought with these other two drugs that what they're probably doing is modulating downstream pathways that may give some clinical benefit. So in the case of the HSP70, it's restoring one aspect of the disease, which is the myelination specifically. But I think that what this shows is the fact that it's very, very difficult to predict. There's still controversy about how miglustats work in Niemann-Pick C. Is it via GBA2 inhibition? There's a paper out there that suggests that that's... So I think empirically they're working, and I think what we've got to do is, personally, I feel that we're not discovering enough drugs in this area because we have a formula about how we do drug discovery. That we think we know the target, we think we can have an assay. But I would argue, based on this, and again, it's taken 30 years it's not a quick process, But if you do a deep dive into these processes and then you try and understand how modulatory drugs that you may be testing for one reason, I mean, for example, acetyleucine was almost an accidental clinical discovery that it was working in cerebellar ataxias. Nobody had any a priori reason to test it. That actually some of this sort of serendipitous discovery, which miglustat was as well, remember we were working on antiviral actions of this, proved it could never work in HIV, and then found that it would work in this rare disease. So I totally agree, Andrea. I think we've got a problem with drug discovery, and it's of our own making. But it's very difficult to get, no pharma companies have got the resident expertise to go into these things in depth for decades. And I would also point out that I can't think, and maybe you can correct me, Andrea, but I can't think of a single lysosomal disease-approved drug in any disorder that was not academically discovered and developed. So I think, again, we have to have these closer ties between academia and industry. And also, I would urge anybody who's in the younger age group in the room is go to cell-based assays as soon as you possibly can. Because with miglustat, you would never in a million years have chosen it as a treatment for Gaucher based on KIs. It was dismissed. It was too weak. But in fact, it works. And it's actually just about the right balance of hydrophobicity. So, yeah, I totally agree. I think we have a problem. And hopefully this meeting today is to, start those conversations. How do we rethink this?
I was just wondering, you mentioned collateral effects in the spirit. Clearly the cell-specific action, dysfunction, such as vertigo, for example. I suffer from the worst form, which got worse with age, and I want to jump. And not only that, I want to tell people and I can't go over that. And that is curiously specific. I just wanted you focused on the lysosome, demonstrated effects. Are there any organelle-specific and cell-specific dysfunctions associated with the lysosome dysfunction?
Yeah.
So we are mitochondria only in a particular cell.
Yeah, so I think that's a really interesting question. So obviously, if you look at any of these lysosomal storage diseases, for the most part, every lysosome in every cell type has the capacity to be affected. So I think something like Niemann-Pick C, where it's this contact site formation, that's probably happening in all lysosomes to some degree in all cell types, but the consequences of that can be very different. So if you look at the neurodegeneration that ultimately causes a lot of the severe disease, it's incredibly patterned. The Purkinje neurons are particularly vulnerable. Microglia are affected in all of these diseases. And one of the things that Emily and I have been discussing is doing more of in the case of NPC, the organelle crosstalk in live, in fixed tissues from the whole organism, with and without these treatments. So the consequences of, Gaucher disease is primarily a macrophage disorder because most cells can cope with the degradative problem except those that have got the burden of an additional phagocytic, clearance of apoptotic cells, for example. So all of these things have cell type nuances, but the underlying problem is in every cell. But also not all cells make all macromolecules. So in the brain, all the glycolipids, for example, are patterned. So that's why you see different cells dying in GM2 than you do in Niemann-Pick C, for example. But what we really want are treatments that are agnostic to that and that will fix any cell type that the drug can access. Because there's also a large peripheral burden, not just CNS disease in these disorders. And if you fix the brain and leave the periphery, you've still got a very compromised life experience. So, yeah, it's complicated.
Is there any indication of lipid makeup of, for example, the endohepatic reticulum?
Well, that would be something. I think with the new technologies of organelle isolation, we're getting a little bit more information on the fine detail of this. But I think I've just did a sphingolipid meeting, Gordon conference, and we still have really poor data on organelle lipid profiles, let alone what's going on in the two leaflets of the membrane, where we know that there's a lot of effort being made making them asymmetric, and we're not sure what's driving that. So we're not there yet. I think some of the imaging, mass spec imaging techniques may help with that, but we have a very, very poor understanding.
I'm sure there will be plenty of opportunities for the other people to ask more questions. So I want to present our next speaker, Professor Andrea Balabio. Andrea is a pioneer really in the field of lysosomal biology and human genetics. His work really kind of shifted our understanding of lysosomes just as recycling organelles into more like signaling hubs. So his contributions have been really recognized by numerous major awards, including the Louis Jeantet Prize of Medicine and multiple ERC advanced grants. He holds leading academic positions across Europe and also the US, including as a visiting professor here in Oxford. Today we'll be speaking on lysosomal signaling and metabolic adaptation and tumorigenesis. And please join me in welcoming Andrea.
Good morning. I'm really glad to be here. I'd like to thank the organizer for the invitation, and particularly my old friends, Fran and Anthony. We have done many things together, working on lysosomal storage disorders and on the lysosome. So, as you heard, in the past few years, I focused my research on these little organelles that are present in all cells of our body, the lysosomes. You can easily visualize the lysosome by immunofluorescence. In this case was a lysosomal membrane protein, just like NPC1. In this case was LAMP1, very abundant protein. So a few years ago, we made a simple hypothesis. We thought that lysosomal function, lysosome number, and the lysosome-mediated degradation process had to be regulated. That makes sense, right? That's a very obvious thought, but that had never been even tested because lysosomal enzyme, genes encoding for lysosomal enzymes were thought to be housekeeping, this horrible term, always on. They didn't need to be regulated. But we thought that everything in the cell should be regulated, for instance, to respond to extracellular cues, environmental cues. So I wish I could tell you how we made this discovery, because it's quite fascinating, but I don't have the time to do it. I just tell you that we did it in silico without doing many experiments, just testing data that were deposited in the databases. We tested co-expression analysis of lysosomal genes and promoter analysis of lysosomal genes. Of course, when I say lysosomal genes, I mean genes encoding for lysosomal proteins. So by doing this, we discover a gene and its protein product that indeed, in specific condition, it's in the nucleus. So also we established like a link, a functional link between the nucleus and the lysosome. So this gene encodes a transcription factor, and the name is TFEB, transcription factor EB, and is now recognized as the master regulator of both lysosomal biogenesis and autophagy. So I guess you all know that one of the main functions of the lysosome also is to mediate autophagy. Autophagy is a process that is mediated by two different cellular organelles. So it's right in the topic of this meeting, the lysosome and the autophagosomes. You can view the lysosome as incinerators, even though I don't view them as incinerators, and the autophagy as garbage trucks. So here is where degradation and recycling occurs, and here is where cargo delivery to lysosome, to the incinerator, occurs. And TFEB can be viewed as a rheostat. So not a switch. So the lysosomes and the autophagosome exist even without TFAB. So you just need TFAB to adapt. So you need TFAB to adapt lysosomal function to a number of stimuli. So when you turn it to high, you have induced lysosomal function, increased level also lysosomal enzymes, increased number of lysosomes. It's a true regulator of lysosomal biogenesis and also of the biogenesis of autophagosomes. So by regulating separate genes, different genes. So now this is a transcription factor, and it belongs to the helix, loop helix, leucine zipper family of transcription factor. You know, I'm sure you know many members of this family, like MINC, MAD, MAX, MyoD, and so on. They regulate many different important aspects of cell function. In particular, TFAB belongs to a subfamily with 4 members, MITF, TFEB, TFE3, and TFEC. So most of the things that I'm going to tell you about TFAB apply also to these other genes and proteins. They can both homodimerize and heterodimerize. They bind DNA as dimers. And they bind similar set of promoters, so there's a high degree of redundancy, and are also post-translationally regulated in a similar manner. I'll show you that post-translational regulation for these proteins, for these transcription factors, is very, very important. So just to give an idea on how broad the control is of TFAB on the lysosomal autophagy pathway, I thought to put some of the targets here, the direct transcriptional targets, but they are not all of them. For instance, here I have lysosomal membrane proteins, and I need to update it because I have to put PMCA2 here, right? So I just when I go back to my hotel, I will add. Okay, so but there's many missing actually. So, but just to give an idea, these targets are in many different steps of autophagosome formation, BPS34 complex, ITG9 complex, cargo recruitment, isolation membrane. So many of the steps for forming autophagosomes are controlled by TFAB. They also control, TFAB also controls these target genes that are involved in autophagosome-lysosome fusion, and of course, for the lysosome, the hydrolases, the membrane proteins, channels and transporters, and also several subunits of the VATPase, which is the machinery responsible for the acidification of the organelles. Now, you don't need to regulate all of the proteins of the lysosome or the phagosome. You just need to regulate the ones that are the limiting factors for the function of the organelle in order to have the increase or the decrease of the function. Now, another important thing I need to tell you, even though it's an old observation that we made, is that TFAB shuttles continuously between the cytoplasm and the nucleus. And now if you grow cells and look at TFAB localization in cells that are normally fed, you would find TFAB in the cytoplasm. So it's actually a surprise because it's a transcription factor. You expect it to be in the nucleus. And the same is with a living organism like a mouse. So you sacrifice the mouse, and in general, you will find TFAB in the cytoplasm unless you starve the mouse or you starve the cells. So starvation is one of the stimuli, one of the main stimuli that promote TFAB nuclear translocation. And there are many others: infections, stress conditions, in the muscle, physical exercise, inflammatory agents, increased calcium levels, and there's like hundreds of different stimuli have been studied, because this nuclear translocation is quite important. But the best studied is starvation. So if you eliminate amino acids from the culture medium, then you will see very rapidly, in less than an hour, you see TFAB nuclear translocation. And it makes sense. Why starvation? Because with starvation, you need to induce degradation processes and cellular catabolism to generate energy. So what's the mechanism for this? The mechanism for this is phosphorylation. So basically, there are some particular serine residues in the TFAB protein that when they are phosphorylated, they retain TFAB in the cytoplasm. And the kinase, the main kinase, there are several, but the main kinase that is responsible for this phosphorylation is mTORC1, which is another master regulator, but in this case, it's a master regulator of cell anabolism rather than cell catabolism. So it regulates protein biosynthesis and consequently regulates also cell growth and cell proliferation. So you immediately realize that this is a switch between anabolism and catabolism. When nutrients are present, mTORC1 is induced, and mTORC1 is known to be induced by nutrient, and it phosphorylates TFAB, and so catabolism is blocked by retaining TFAB in the cytoplasm. When mTORC1 is inhibited by starvation, then TFAB translocates into the nucleus, and protein biosynthesis is inhibited, and then lysosomal and autophagy function are induced. So it's a main switch for cell metabolism. And this is the normal physiological situation, and then I will show you some aberrant situations. Now, we also discovered that TFAB, when it is in the nucleus, is able to hyperactivate mTORC1. So if you overexpress TFAB, you see a hyperactivation of mTORC1. And this is a feedback loop, because, of course, the more mTORC1 you have, the more TFAB is blocked and cannot do this anymore. But in the case of aberrant situation, you will see when this is disrupted, then you will see that sometimes TFAB constitutive activation or hyperactivation is also associated with mTORC1 hyperactivation. But this is an abnormal situation. Okay, just to give an idea on how important is mTORC1 for TFAB subcellular localization, I'm going to show you this simple experiment. We treated HeLa cells that are stable transfectant with TFAB GFP. You see that we treated them with an mTOR inhibitor, Torin-1, a catalytic inhibitor of mTOR. You see, before treatment, TFAB is localized in the cytoplasm and also on the lysosome because it is phosphorylated on the lysosome by mTORC1. I will expand on this because that's a very important concept. So mTORC1 to be active needs to be recruited to the lysosomal surface. And there is where it meets TFAB and it phosphorylates TFAB. So basically now the lysosome has a way to regulate its own function by hosting mTORC1 on its surface. So when you start the treatment, this is what happens. You see that after, in this case, 120 minutes, 2 hours, TFAB is completely nuclear. It stays also on the lysosomes, but it disappears from the cytosol. So that also tells you that one, in principle, could modulate TFAB function with a drug. Now, this is a toxic drug, Torin. It's a very potent inhibitor of mTOR, so it cannot be used in the clinic. But, you know, rapamycin can. Unfortunately, TFAB is a rapamycin-insensitive substrate, mTORC1 substrate. But in principle, one could do that. So a switch between anabolism and catabolism, and I'd like to emphasize that this switch occurs on the lysosomal surface. That's a little bit a revolution about the view of the organelle. It's not only an incinerator or a garbage can, but it's actually hosting processes that mediate not only cell catabolism, but also cell anabolism, and in general cell metabolism in response to nutrients. So So a new view of the lysosome, a new concept is lysosomal regulation. Lysosomal function is subject to a global control. Then another new concept is lysosomal signaling. The lysosome acts at a signaling hub. mTORC1 is, and now there's many other kinases that are known to be recruited on the lysosomal surface and to be activated on the lysosome. Actually, by the way, this is not our discovery. This was the discovery of David Sabatini. Pretty much at the same time when we discovered TFAB, we discovered that mTORC1 to be active needed to be recruited on the lysosomal surface. And then the other concept is lysosomal adaptation. So the lysosome has the ability to sense cellular environment, and the main sensor is mTORC1, and to adapt its function to cellular needs. And in this case, the adapter, the mean for adaptation is TFAB. So more recently, we found that mTOR can be substrate specific. So I'm just going to show you 2 slides on this, even though this was, I think, a very interesting discovery and also unexpected, because people thought that mTORC1 was either active on all substrates or inactive on all substrates. And in fact, people measuring mTORC1 activity in thousands of papers, people were using one substrate, S6 kinase, or maybe 2 substrates, S6 kinase and 40BP1, and would say, oh, mTOR is active, or mTOR is inactive. But nobody actually realized that mTOR could be, in some conditions, could be active towards some substrates and inactive on other substrates. And so we basically discovered what we now call canonical, that the canonical pathway requires Reb as an activator of mTOR, and Reb was described as a protein that is absolutely needed for mTOR activity. But we also found a non-canonical branch of mTORC1 signaling that doesn't need Reb, it doesn't need growth factor, it only needs amino acids, and Reb is dispensable for this non-canonical branch, but this branch needs this gap GTPases folliculin and its partner FNIP. So that's the canonical for canonical subset, and the non-canonical is for TFAB and the other member of the TFAB family, of the MITTFE family. So in some situation, mTOR could be active on TFAB, for instance, and not on S6 kinase and 40BP1, and also there are some opposite situations. Now, people were not entirely convinced, some people, but to convince people you need to show structural evidence. So we entered into a fantastic collaboration with the group of Jim Harley and Lucas Huber. Jim made the cryo-EM, we did the functional analysis of this, and this is the cryo-EM structure of what we call the mTORC1 TFAB drugs regulator megacomplex, 36 polypeptides, and this is TFAB here in yellow. You can see it from the other side, and it's right where we predict it to be between the RAG GTPases, and you could view also the non-canonical branch and the canonical branch they coexist in the same structure. So I thought it was very, very beautiful, so I tried to convince my wife to put this in the living room. No way. Too many colors. So then I pushed it. Maybe we could reduce the color, and that what we agreed upon. This. So I was able to retain the color only for TFAB. Okay, so can we modulate TFAB activity for therapeutic purposes? That's the translational part. When we made this discovery, we and also many other groups realized that this could be a therapeutic tool, Why? Because it induces intracellular clearance by inducing lysosomal biogenesis and autophagy. So one could clean the cells by activating this pathway. And the beauty is that you need to do it on only one protein, because the one protein controls all the others. So that was very attractive. And so we and others did it in many different types of diseases. So for instance, we did it in lysosomal storage disorders. Other people do this. Each of these is a separate paper, but there are hundreds of papers. So people did it on neurodegenerative diseases. There are only on Parkinson, there are about 50 papers on Parkinson with this approach, and showing clearance of accumulation of alpha-synuclein and rescue of the disease phenotype, Huntington disease, Alzheimer's disease, clearance of tau proteins, and even metabolic disorders like diet-induced obesity is responsive to TFAB activation. Actually, I'm going to show you this example. These are the liver of normal mice, and this is the oil red staining for lipids. This is a normal mouse fed with the IFAD diet. You see lipid droplets and oil red staining. This is a TFAB knockout, conditional knockout mouse in the liver. You have more lipid droplets, more lipid accumulation. And this is a mouse infected with a virus, like a gene therapy approach using TFAB. You have almost complete rescue of liver size, HE staining, so no lipid droplets anymore, and very little oil red stains, and these are the quantification. And what's interesting is that these mice treated with TFAB, they don't eat less. They actually eat more, but they are much leaner. And you see they lose weight even with late treatment. So very attractive, but there is a but, because being this a major regulator of cell metabolism, you can't really activate this permanently. Otherwise, you're bound to problems, and the problems are linked to cancer. So, in fact, members of the MIT TFE family, like TFAB and particularly TFE3, a brother of TFAB, have already been described as oncogenes, so associated actually with renal cancer, with the chromosomal translocation leading to overexpression of these genes in the liver and causing renal cell carcinoma. We actually focused our attention more recently on a disease called Burgh-Dubé syndrome, which is an inherited cancer syndrome, which is also associated with kidney tumors, but also pulmonary cyst and benign skin tumors. And this is why we became interested, because this disease is caused by germline loss of function of the folliculin gene, which is folliculin is a protein that is necessary for the mTORC1-mediated phosphorylation of TFAB. So without folliculin, TFAB cannot be phosphorylated by mTORC1, and it's always nuclear. And so TFAB is always nuclear in this situation. Now, to get the kidney tumor, these patients also have somatic mutations leading to loss of heterozygosity. So they actually lack folliculin function in the kidney when they develop tumors. So that's folliculin. That's required for mTORC1-mediated phosphorylation of TFAB. When you don't have folliculin, not only you have TFAB constitutively active and nuclear, but also you have this feedback loop that is disrupted. And so you have TFAB active, always in the nucleus, and mTORC1 hyperactive because of what I told you before. TFAB can hyperactivate mTORC1. So you have both cell anabolism and cell catabolism hyperactive in the same cells, basically. And that's really bad. Actually, it's good for the cells because they grow more, they proliferate more. But it's bad for the organism because then you get cancer. And that's the main cancer driving mechanism. I'll show you what Chiara Di Malta, very talented postdoc now, independent investigator, did in the lab. So first of all, she observed, of course, that with follicular knockout, TFAB is always nuclear. Also, in the presence of nutrients, you see it's nuclear. The control is cytoplasmic, and then follicular knockout is nuclear. This is a follicular kidney-specific knockout mouse model in which you have a very aggressive phenotype. The mice die because of cystic and tumorigenic phenotype. And then when she did the double knockout of folliculin and TFAB, she observed a total rescue of the disease phenotype, rescue of the lethality. All these mice die because of renal insufficiency before 30 days of age, and these have normal survival, normal renal function, so they're totally rescued. And then after our discovery, Lisa Henske at Harvard asked for our TFAB knockout mice, and she crossed them with another a mouse model of another inherited cancer syndrome, which is tuberous sclerosis. And then when she did the double knockout, also total rescue of the disease phenotype. So that's an important tumorigenic mechanism. So is it possible to pharmacologically modulate the TFAB activity? I'll keep it short. So, because, what do we want to do? We want to activate TFAB to treat neurodegenerative diseases, for instance, and you want to inhibit TFAB in the context of cancer. Now, keep in mind that these phenotypes that I showed you are permanent modifications. Now, the pharmacological treatment is a pulsatile modification, and so we believe that with pulsatile modification, you will not get the phenotypes that we saw with genetic permanent modification. So, to keep it really, really short, we developed a TFAB responsive transcriptional reporter by using a promoter of the most responsive TFAB target, which is GPNMB. So we took the promoter of GPNMB and we put it in front of mCherry. And so this is a very sensitive reporter. When you overexpress TFAB, you see mCherry appearing, very low background. And also in follicular knockout and TSC2 knockout cells, you see mCherry very much induced and little background in the wild-type cells. So we have been using this reporter for drug screening, both for induction of TFAB and inhibition of TFAB, using a number of different libraries. And I want to show you just the results with this compound here, E22, that we identified by using this screening. So we tested it on spheroids, and you can see that very effective in the spheroid growth for a tumor from a TSC patient, a tumor from translocation, renal cell carcinoma, chromosomal translocation, and a chromophobe tumor from a patient with the BHD syndrome, the syndrome that I will tell you before. And you see that very effective in delaying and diminishing tumor growth. But we also tested this in vivo, and you see the results are really encouraging. This is untreated, treated with vehicle, and this is the compound. Very much reduced cystic and tumorigenic phenotype, and also we tested TFAB, TFE3 levels, and the mTORC1 activity, and they return actually almost to normal levels. And GPNMB is a TFAB target, and these are the quantifications. So we are now focusing on this compound, and we have several other candidate compounds. And I'll skip this. So conclusions. The function of two cellular organelles, lysosome and autophagosome, can be modulated by acting on a single protein, TFAB, master transcriptional regulator. Some diseases, neurodegenerative diseases, for instance, would benefit from TFAB induction, whereas other cancer from TFAB inhibition. Permanent induction or inhibition, e.g. by genetic approaches, of TFAB would be bound to severe side effects, suggesting that a pulsatile pharmacological approach should be used. So, for instance, in disorder characterized by accumulation of substrates, like neurodegenerative diseases, you want to induce the pathway until you clear the cells, and then before you give it another shot with a drug, you wait until the accumulation builds up again. And we believe that in this way you avoid the cancer, the effect on cell growth and proliferation. And then the other thing is that I showed you is that TFAB activity can both be induced and inhibited pharmacologically. It's just a matter of finding the best drugs, which is not trivial, of course. Thank you. As you can see here, many collaborators, very important collaborators.
Thank you, Andrea. We have time for one quick question. Yes,
yeah, you introduced the whole family of helix to helix transcription factors, then you really focused on TFAB. What do the other ones bring to the whole equation? Are they subtype selective? Are they alternative targets for the approaches you just highlighted?
Okay, so there's a big family and a smaller family. The big family HLH using zipper. There is some crosstalk with MYC. So MYC is able to compete with some of TFAB binding sites. Now, it's a completely different story for the subfamily, so the smaller family, MIT-TFE family. So there's 4 proteins, TFAB, TFE3, MITF, and TFEC. So nobody has studied TFEC because it's expressed at very, very low levels, and nobody really knows its function. The other 3 are quite redundant. So MITF is a master regulator of melanosome biogenesis. But if you use overexpressed TFAB in melanocytes, it becomes also a regulator of melanosome biogenesis. So it's a matter of in what cell types are expressed. So I showed you that by knocking out TFAB, you rescue the phenotype. But just because, so what about the other 2? Now, TFAB is much, much more expressed in the cell type where we knocked out the gene, which are the cadherin 16, because we use a cadherin 16 CRE. But if you use another CRE, then TFE3 becomes more relevant. It's just a matter of where they are expressed. They bind, we and many others found that they bind pretty much to the same promoters. They have the ability to bind to the same promoters. And they're also regulated by mTOR in the same way. The serine are conserved. And so what is really different between them is the expression level in specific cell types. Now, of course, there's possibility of also interactions with other proteins that could be different, but nobody actually has come up with a convincing result on this.
Thank you very much.
Yeah.
I'm afraid we have to move on. So now we have one short oral communication. Unfortunately, our second speaker had to withdraw due to health reasons. So we have Maggie Moore coming. Yeah.
Am I going the right way? Okay. No.
Yeah, keep going, keep going.
That's yours, isn't it? Yes. And it's that one there.
Yeah, okay. Thank you. So, hi everyone, my name is Maggie, and I'm a PhD student from Professor Sander Patel's lab, and today I'm going to present the remote control of ion selectivity switching in a lysosomal ion channel that tempers the calcium permeability. So lysosomes is most well known for its function as the recycling center in the cells. However, it is also a very important intracellular store for the calcium ions, besides the endoplasmic reticulum. And there are various ion channels that regulate the calcium release, and today I'm going to focus on a very interesting channel, called two-port channel, TPCs. And TPCs are an ancient member of the voltage-gated ion channel superfamily, and they are expressed predominantly on the acidic organelles, such as endosomes and the lysosomes, where they regulate the trafficking of various cargoes, and they are also implicated in a range of diseases. However, despite such pathophysiological importance, their activating ligands and the selectivity was under debate for decades. And a few years ago, there have been two very important findings that form the basis of my study. So surprisingly, the TPC2 can switch its ion selectivity in a ligand-dependent manner. So when they are activated by the calcium mobilizing messenger ADP, they act as a non-selective cation channel with a robust calcium signal. And when they are activated by the regulatory lipid PIP2, they act as a sodium selective channel. And a few years ago, two synthetic agonists called TPC2A1N and TPC2A1P had been identified high throughput screening by Christian Grimm's group, and they mimetic the effects of ADP or PIP2 respectively. And this has allowed a selective activation of the channel, and therefore a more detailed study of the TPC2 in vivo and also in intact cells in a lot simpler and faster way. And despite that, ADP and PIP2 have dramatically different effects on the channel activation, rendering the channel either more calcium permeable or more sodium selective activation. A few years ago, a recent work from my colleague has discovered that the channel actually can be co-activated by ADP and PIP2, and as well as their mimetics, to markedly potentiate the calcium but not the sodium permeability, and revealing a synergistic regulation of the channel. And my question is, what's the mechanism behind it? So the Quail-EM study have reviewed the structure of human TPC2, and PIP2, which binds the channel directly, has its binding site identified on the S4-S5 linker in the first domain, which is clearly very important for the channel activation. So I did an alanine screening along the S4-S5 linker where the PIP2 binding pocket locates. I mutated the residual from 212 to 218, and the screening is coupled with another mutation at L11 and L12. This is to reroute the channel from the lysosomes to the plasma membrane. And so I first started with investigating the A1N-induced calcium response in PM-targeted TPC2 channel in HeLa cells loaded with the fluorescent dye FURA2. So the traces are not working properly here because of the graph prism issue. And the N compound has triggered a prompt calcium signal in the wild-type PM-targeted wild-type channel. And interestingly, all the mutants had various effects on the calcium response. So two of them have blocked the A1-induced calcium response, and S218 had a little effect on it. And interestingly, we had found two gain-of-function variants, P214A and E215A. And E215A is the most potent one here, so it will be the focus in my following research. And this is the first take-home message, that the alanine scanning of the S4-S5 linker had identified a gain-of-function mutant. And so therefore, I have next extended my study to a 1P compound with the E215A mutation. So in the wild-type channel, the P compound gave us recordable but a delayed calcium response, and which has been massively enhanced with the E215A mutation. And now it looks similar to the calcium response induced by the A1N compound. And then our collaborator, Tofik from Cambridge, has done electrophysiology in TPC2 in macropatches excised from the plasma membrane. And here you can see in the wild type channel, the N compound in blue trace here give us mainly inward calcium current, while the P compound give us predominantly a sodium current. And however, with the E215A mutation, the calcium response has been potentiated for both N and P compound, and now the response of two agonists looking similar to each other. So the second take-home message is that this gain-of-function mutant has reduced agonist-dependent changes in the TYP2 permeability. And then we have looked back to our old published data where, of the agonist combination in the wild-type channel. And it seems that the E215A is phenocopying the combo effect in the wild-type channel. So I started to wonder what would happen if I add both agonists to this mutation. And this time I have used a lower concentration of both agonists. So with a lower concentration of agonists, in the wild-type channel, the N and P compounds have evoked a relatively mild calcium response, and when they're added together, this response has been largely synergized. However, in the E215A mutation, the N and P compound have both largely potentiated this agonist-induced calcium response. However, when they are added together to this mutant channel, the synergistic effect has been reduced, and in another word, they have now become additive. And I would like to point out that a naturally occurring disease, TYP2 mutant, found in an albinism child, have been reported to be a gain-of-function. And interestingly, this mutation R210C is also in the proximal S4, S5 linker of the channel, where the PIP2 binding site locates. And I hypothesized that this mutation also perturbs the agonist action in a similar way to E215. So I have next tested the effects of NMP both singly and in combination to this mutation R210C. And in the cells expressing R210C mutation, the calcium response induced by A1N and P in blue and orange traces here are massively enhanced compared to the wild-type channel. However, when the N and P are added together, the calcium entry is not further accelerated relative to either agonist alone. And therefore, I concluded that the R210C mutation had again disrupted the agonist, disrupted the synergistic regulation of both agonists, similar to E215A. And I would like to mention that the basal calcium level of this mutant is also elevated, and this points towards a constitutive activation of the channel, and which is consistent with what's described in this literature. And so, therefore, my third take-home message is that this gain-of-function not only reduced the agonist-dependent changes in the TYP2 permeability, but it also reduced the synergistic activation of the TYP2 by its agonists. And then our collaborator, Philip from Oxford, had done the molecular dynamic simulation and had identified the residual interaction of E215. So E215 potentially interacts with Q214 in S6 domain in the first repeat of the channel, which is where the activation gate locates. And I have therefore studied the Q214 mutation with NNP and a combination of both. So in the cells expressing the Q214, the N and P calcium signal, sorry, the trace disappeared from here. In cells expressing Q214, the N and P calcium signal have been largely potentiated. However, when they're added together, they are, the calcium response is only modestly increased compared to the wild type. So again, similar to E215A, the Q214A has also reduced the synergistic regulation of this channel. So the fourth conclusion is that the interaction between the linker and the gate also regulate the agonist selective modulation of TPC2. And so far, all my experiments have relied on TPCs that being rerouted to the plasma membrane. So I have next analyzed the TPC in their natural localization, which is on the lysosomes. So in the lysosomal TPC2 channel, the N compound gives us a modest calcium signal, which has been largely potentiated with the E215A mutation. And the P compound gives us almost no response in the wild-type channel, and it is again largely increased with this E215A mutation. And therefore, we found that the deregulated agonist activation of TPC2 also perturbs the lysosomal calcium flux. And following this observation, we would like to assess the E215A mutant in vivo. And to do this, Francesca and Kyle from our lab has worked with Eric Lemby, and they used a C. elegans as a model system. So C. elegans, they lack a functional TPC2 ortholog, so therefore they can effectively serve as the TPC2 natural TPC2 knockout. And taking advantage of this, they have introduced the human TPC2 under heat shock promoter, and allowing inducible and ubiquitous expression of the TPC2 upon heat stress. And they have first assessed first performed the locomotion assays on to assess how the expression of the wild-type TPC2 and E215A affect the adult worms. So here you can see the worms before the heat shock when the channels are not yet expressed. And here is the worms after heat shock when the expression is induced. So in the wild-type channel, the locomotion seems unaffected before and after heat shock. And in contrast, in the worms expressing the E215A, the movement are normal before the heat shock, and however, all the worms becomes completely paralyzed 1 hour after heat shock when the channel is highly expressed. And the quantification have showed a very significant and time-dependent paralysis of the worms that express the E215A mutation. And however, the movement is not affected in the worms expressing the wild-type channel. And they have also assessed the survival of animals 24 hours after the heat shock. You can see, and we have observed very significant reduction of the viability of worms with the E215A mutation, while it is unaffected in the wild-type with the wild-type channel. And they have also generated a double mutant worm harboring both the gain-of-function mutation and the L265P, which is a poor dead version of TPC2. And in this double mutant, the viability of worms is also unaffected, and this has proved that the toxicity is dependent on the channel activation. So the sixth conclusion is that the deregulated agonist activation of TPC2 perturbs locomotion and viability in vivo. And most importantly, with one single mutation, we have turned TPC2 with very highly unusual channel behavior into a regular channel, where both agonists have more or less the same effects. This is remarkable given that the both agonists have normally have very dramatically different effects. And I would like to thank our lab and our collaborators, and thank you for listening. I'm open to questions.
Any questions? I actually have one. So you said that documentation that almost renders the TPC2 as constitutively active. Do you know if that has repercussions, for example, the resting pH of the lysosome?
I'm not sure it's directly linked to the pH. So
So you haven't tried, because obviously you know that the calcium flux also has like the resulting changes in pH when TBCP is activated. So if you've got that hyperactivation channel in the lysosome, would that affect the pH and then the functions that are regulated by lysosomal pH?
Yeah, I think so. Adding the drugs A1N would increase the pH in the channel. I'm not sure it's like directly linked to the constitutive opening of the channel of the arginine C, but I think arginine C, I think they cause the hyperpigmentation in the patients because they regulate the pH of the melanosomes and something like that. So, that's fine.
Thank you.
Yeah.
Any other questions? Yes.
Everyone, there's an emerging topic in lysosomal biology, which is the response to disturbances in the homeostasis of calcium and also the homeostasis of protones or pH. It's a sort of lysosomal stress response. There's many papers now on this. And you can easily check if that's maybe I'll talk to you later. There are some simple experiments that you can test with your mutant and with your channel to see if that's also involved in this lysosomal stress response. It's a recruitment of kinases and several downstream it's called CASN, C-A-S-N. And it's also linked to LC3 lipidation on single membrane rather than double membrane like so they are LC3 regulated in lysosomes. So I think it would be very interesting to test that.
Yeah, sure. Thank you.
So our next speaker is Dr. Organelle Pharmacology. Nuno's research focuses on communication between organelles. Again, carrying on the theme from the presentation before. And he aims to understand how this crosstalk contributes to aging and diseases such as neurodegeneration and cancer. So today, you'll be speaking on targeting signaling elicited by mitochondrial and lysosomal effects. Welcome.
Thank you very much. Good morning. Thank you very much to Anthony and Fran for the kind invitation to be here. It's really a pleasure. We don't really target the actual defects like Fran showed beautifully, but we are working our way upstream the chain. This is how we see our cells. It's a complex system of multi-components that are carefully coordinated with each other. Doesn't mean that we know where they are coordinated and how they are coordinated, but we assume that everything inside the cell is coordinated. Every event is coordinated with another. And of course, the corollary of the previous slide is that in order to understand a biochemical defect and its consequences for pathology, we need to understand not only what's wrong with that particular component, but how the defect in any of the components that are affected in the cell affect other components and the whole cell as a system. Whenever there is a biochemical defect caused by a gene mutation, for example, of course, that gene is affected, and if that gene happens to encode for a protein that is inside an organelle, that organelle will also be affected. And then the cells are going to sense that there's something wrong with it, either because there's a metabolite accumulating or because there's a metabolite missing where it needs to be, and they are going to respond to it. And that response can be an adaptation or can be a loss of the cell. When cells adapt or when they die, that changes the way that particular cell that was affected communicates with other cells, which eventually will result in the pathology that we recognize, organs malfunctioning and symptoms being presented in the clinics. Of course, the more upstream we go on these scheme, the closer we are to the root of the problem and the better we are able to deal with the situation that is occurring in the patients. Unfortunately, most of the times, and in rare diseases at the moment, we are right down here, the treatment, the clinical strategy is just symptomatic. The clinicians try to deal with the symptoms that the patients present because there is no way to deal closer to the root causes of the diseases. We started applying this premise a few years back in the context of mitochondrial diseases, which is a group of about 300 diseases that are caused by different mutations in either nuclear encoded genes that generate mitochondrial protein products or in mitochondrial DNA itself. These diseases affect predominantly the central nervous system, the skeletal muscle, and the heart. They have a very marked tissue specificity, and there is no cure for these diseases. Again, it's symptomatic clinical management. Whenever there is a mitochondrial disease, there is always the explanation or the tentative explanation that this disease is arising because mitochondria are not doing well, therefore ATP is not being produced in enough amounts, and that causes the disease. This is a very nice explanation. There's just one problem with it. It's not true. We have worked with several models of mitochondrial disease, and we have observed that if we block the signaling cascades that are elicited by the mitochondrial defect, even if the mitochondrial defect is still there, we can remove the pathology from animal models in vivo and, of course, in cellular models in vitro. So this tells us that it's not the energy inability of deficient mitochondria, that it's not the lack of ATP that is causing disease, but it's the signaling that is elicited by these organelles that is leading to a cascade that eventually results in pathology. This signaling can be activation or manipulation of signaling cascades in the cytoplasm, but it can also be modulation of the function of other organelles inside the cell. I don't need to preach to the converted. Anthony already alluded to these. There's many instances of organelle communication between mitochondria and the endoplasmic reticulum via contact sites. For example, mitochondria and peroxisomes share metabolic pathways such as fatty acid oxidation, antiviral signaling proteins, fission mediators. The peroxisomal biogenesis does not occur if the mitochondrial respiratory chain is not functional, and the respiratory chain cannot assemble supercomplexes if the peroxisomes are not functioning, because an essential lipid to allow the formation of these supercomplexes is generated exclusively by the peroxisomes. Obviously, the lysosomes, as Andrea nicely pointed out, are necessary for the autophagic removal of other organelles and structures in the cell. Mitochondria release vesicle traffic that is directed either to peroxisomes or to lysosomes, and the contents of these vesicles are highly controlled. And mitochondria also have contact sites with the lysosomes, as Emily nicely introduced. This is a pretty mitochondrial-centric view of things, but the lysosomes also communicate with the nucleus, as we heard from Andrea, in which the transcription factor TFAB goes to the nucleus and drives lysosomal biogenesis, and we've recently found that in some lysosomal diseases there is defects in nuclear DNA maintenance, and the lysosomes are actually actively contributing to this process. So whenever we look at different organelles communicating with each other, that provides us with an opportunity. Sometimes, and oftentimes I would say, whenever there is a mitochondrial disease, the lysosomes are also dysfunctional. Whenever there are lysosomal storage diseases, mitochondria are usually also dysfunctional. This means that if we can't treat the mitochondria in a mitochondrial disease because there's a defect in somewhere in a subunit in the respiratory chain and we cannot get to it, perhaps by modulating lysosomal function and by improving lysosomal function in these cells, these models, or in these patients, we can at least alleviate some of the symptoms that we are observing. And we've demonstrated that that's possible using cellular models of cellular and in vivo models of mitochondrial malfunction, in which by modulating lysosomal behavior, particularly through the MCOL1 or TRPML1 channel, we can rescue some of these phenotypes of mitochondrial deficiency. We've also observed that in some lysosomal defects that result, for example, in functional iron deficiency due to storage of iron inside the lysosome, and lack of it, therefore, in the cytoplasm. If we supply iron in a way that it's independent of lysosomal trafficking, we can rescue in vitro and in vivo some of these phenotypes. And tomorrow, Nilsa will present a recent work that we've been exploring in which lysosomal defects result in DNA damage, and these triggers the activation of the c-Abl YAP1 signaling cascade, and by which leads to apoptosis, and by inhibiting c-Abl, we can avoid the pro-apoptotic signaling. So again, we are not fixing the primary biochemical problem. The lysosomes are still dysfunctional, but if we don't allow the cells to respond to that, there are some benefits that can be exploited. But today the story I want to tell you about, it's a new project in the sense that we've never talked in public about it, so you are the world premiere. On the other hand, it's a project that we've been developing for about 8 years in collaboration with Ina Milosevic's lab. It started when both of our labs were still in Germany. And recently we've been pushing through and developing it further. Menakshi in Ida's lab, she has a poster tomorrow, and I really encourage you to visit it for more details. It's about a protein called rapconnectin 3 alpha. Don't be fooled by the length of the name. The length of the protein is even worse. It's a monster protein of 330 kilodaltons. It's a nightmare to clone and to work with. And it has been implicated in the regulation of the disassembly cycle of the lysosomal VATPase. So the lysosomal VATPase has these VO and V1 domain, and they routinely disassemble and reassemble, and rapconnectin 3 alpha has been implicated in this cycle. It has also been implicated in several diseases, but the fact is that we still don't know exactly what it is that rapconnectin 3 alpha does. The role of its role in this disassembly cycle of the VATPase has also been shown to be carried out, to be redundant to another ortholog of rapconnectin 3 alpha, which is encoded by DMXL1. Rapconnectin is encoded by DMXL2, so I'll refer to the knockouts as DMXL2 knockouts. And DMXL1 is an ortholog that seems to have similar roles in the regulation of the VATPase. As we will see in a few minutes, these redundancy stops in many of the phenotypes that we are looking at. So one of the reasons we are particularly interested in this protein, besides the clinical phenotypes that arise from its mutations, is that the VATPase is, as Andrea nicely pointed out, at the core of a very important axis in the cell. At the surface of the lysosomes, the cells determine every microsecond, every femtosecond, if they are in an anabolic stage or in a catabolic stage. That's the decision engine of the cells. And this decision depends on the VATPase. In the interaction of the VATPase, we have the proteins like exin, like folliculin, and AMPK, the catabolic kinase, the grand regulator of catabolism, or mTORC, the grand regulator of anabolism. Okay? In a metaphor, this is the person in the government, the minister in the government that promotes building new airports, roads, and infrastructure, and this AMPK is the chancellor. Says there is no money, nobody builds anything. So the balance between these two axes determines everything, or almost everything, that is happening inside the cell. Rapconnectin mutations result in a series of, it's of course a very rare, they are obviously very rare, but they result in a series of related intellectual disabilities that Ida's lab is particularly keen to explore. We are more interested in the aspects of the role of rapconnectin in metabolism. So how do we study this monster protein whose function remains poorly understood? Ida's lab generated a knockout model based on the Cre-LOX system. We are using the MAFs, the fibroblasts of these mice, to do most of the work. It also uses neurons with the knockouts to explore some of these questions. A very important detail that I must point out is that while we take the DMXL2 or RAP connectin 3A from these cells, otherwise it would be a knockout and I wouldn't be telling you this. DMXL1, so the ortholog, is still there and it's there at absolutely normal levels. We don't see any loss of DMXL1. As expected, right? Because we can control this through the Cre-LOX system. But the important part of this is that anything that I show you on the DMXL2 knockout has normal levels of DMXL1, and therefore any phenotype that we observe cannot be attributed to redundancy or to lack of redundancy. It can be attributed exclusively to DMXL2. So we, because of the role that, or the implication that this protein is involved in the regulation of the VATPase, we first looked at lysosomes in these cells by labeling them, by expressing LAMP1 or RAP7, GFP and then Cherry respectively. We can see that the lysosomes in the RAP connecting knockout cells are swollen and bigger. The lysosomal mass is overall increased, and we can see that both by imaging and by the levels of LAMP1, a good marker, one of the most abundant proteins in the lysosomes by Western blot. Because typically increase in lysosomal size denotes a deficit in lysosomal function, we looked at lysosomal acidification, or to the ability of the lysosomes to acidify, and we see that the MXL2 knockouts, they have a higher lysosomal pH. They are not neutralized, they are just not as acidic as the controls. We then started looking at how these cells behave in a little bit more detail, and one of the first things that we noticed, because it was very obvious, is that when we grow the cells in high glucose medium, like everybody uses, like we use routinely in the lab, the DMXL2 knockout cells had a growth deficit. But if we removed glucose from the medium, they grew exactly at the same speed as the control wild type cells. So we thought that this happens because they are not able to uptake glucose. We were wrong. It's exactly the opposite. They uptake about 2 times more glucose than the wild type cells. So if they uptake glucose, but they are not growing in the presence of glucose, we thought that perhaps they are not able to metabolize it. And that turned out to be the case. We looked at the expression level of several, at the protein level of several glycolytic enzymes, both early glycolytic and late glycolytic. And what we observed is that there was a general downregulation of the protein levels of these enzymes in the DMXL2 knockout cells, not only for glycolysis, but also for other pathways like the pentose phosphate pathway that depends on glucose, was also downregulated. Because the inability to metabolize glucose is one of the canonical signals for cells to activate AMPK, the catabolic regulator, we thought that perhaps there is an activation of AMPK in these cells. So we did an experiment in which we moved the cells from high glucose to low glucose or to no glucose. So we removed glucose from the medium. And then we look at the phosphorylation of AMPK substrates. The ones that are shown here is Raptor in serine 792 or ULK1 in serine 317. You can see that the phosphorylation of both of them increased very strongly once we remove glucose from the medium in the wild-type cells, but that did not happen in the DMXL2 knockout cells, suggesting that these cells are not able to metabolize glucose, but they are also not able to mount a glucose starvation response due to the lack of RAP connectin. Because what activates the mechanism that activates the AMPK is intrinsically regulated with the mechanism that also regulates mTORC, as I said, at the lysosomal surface and in a manner dependent on the VATPase. We thought that if the cells are not able to activate the AMPK, perhaps that's because of mTORC. So we went to look at mTORC activation levels. As Andrea pointed out a few minutes ago, P70S6 kinase is one of the substrates of mTOR. And we see that in basal conditions, it's very poorly phosphorylated in the DMXL2 knockout cells, suggesting that the pathway is less active. We didn't look at the phosphorylation of TFAB, but we see that lysosomal biogenesis is upregulated, suggesting that TFAB is active and therefore in agreement with the lower activity of mTOR globally and not just for some of the substrates. So these cells, they are not able to metabolize glucose. They are not able to mount a glucose starvation response, and they are not able to mount an anabolic response. They seem to be extremely confused regarding their metabolic roles. Because mTOR was downregulated at basal level, we sought to test if it's just at basal level or if it has to do with a more dynamic inability of activate mTOR or not. mTOR responds very rapidly to changes in nutrient availability. So we did a classic assay that was published and established by the Sabatini lab 15, 20 years ago, in which we remove amino acids from the medium for about 2 hours, and we see a complete ablation of mTOR signaling by absence of phosphorylation of its target. And then we refeed amino acids for about 30 minutes, and we rapidly see a reactivation of mTOR. Importantly, if this refeeding of amino acids is made in the presence of bacillomycin or any other inhibitor of the VATPase, that reactivation of mTOR is not complete. It's only partial. And you can see here that we refeed amino acids, reactivates mTOR, but if we do that in the presence of bacillomycin, the reactivation is not as extensive. The quantification is illustrated there with a heat map. So this happened in the wild-type cells, as expected. However, in the knockout cells, when we refeed amino acids, we have a recovery of the mTOR signaling as expected. But to our surprise, when we refeed in the presence of bacillomycin, we have an even stronger increase in the mTOR activity. We thought to validate this result, we thought that we would test if this is due to the ability of mTOR to be recruited to the lysosomes where it's activated. And we observed, we did the same experiment, but but monitoring the positioning of mTOR relative to the lysosomes using LAMP1 as a marker for the lysosomes. And what we observed is that when cells don't have amino acids, mTOR is inactive, it does not get recruited to the lysosomes. We refeed, it goes there. But if we refeed in the presence of bacillomycin, the recruitment is not as efficient in the wild-type cells. However, in the knockout cells, the recruitment to the lysosomes is independent of the lysosomal pH. Because treatment with bacillomycin, or if you prefer, inhibition of the VATPase, seemed to be beneficial for the mTOR activity in these cells, in the DMXL2 knockout cells. We decided to test if this was in any way affecting the glycolytic enzymes in these cells. So Meenakshi did a series of painstaking experiments in which she was giving a time course of bacillomycin treatments for us to try to find the sweet spot. And eventually, we determined that about 24 hours of bacillomycin treatment, we could see a low concentration bacillomycin treatment, I must add. We could see an upregulation of the protein levels of most glycolytic enzymes in the wild-type cells, but also in the knockout cells. And while in the clinics, it's not advisable to give bacillomycin to patients. It's quite common to give chloroquine to patients, and it's used in many aspects in malaria treatment, for example. And chloroquine also has the effect that it neutralizes the lysosomal pH. So we tested, or Meenakshi tested, the premise that if we treat those cells with chloroquine, perhaps it's somehow beneficial to the phenotypes that we observed in the DMXL2 knockout cells. And again, we observed in high glucose a deficit of growth in these cells, but if we treat them at the same time with chloroquine, they grow exactly at the same speed as the control cells. suggesting again that if we restore glycolytic enzymes, that is beneficial for these cells. Because mTORC1 signaling was downregulated in these cells, and mTORC signaling is targetable, it's a kinase, there's many ways to manipulate the activity of this enzyme. One of them is by reducing the levels of one of its inhibitors, TSC2. When we silence TSC2 as an attempt to increase mTORC activity, we see that the levels of glycolytic enzymes also increase both in the wild type and in the knockout cells. Again, the quantification is illustrated there. And this is a proof of principle that if we manipulate mTORC signaling in these cells, that can have benefits for the behavior of the cells. Of course, we cannot go and give siRNA to the patients, it's frowned upon. But there are small molecule mediators that allow us to manipulate mTORC activity. And Meenakshi tested some of those in a relatively short time frame. And as we can see here, for example, this is an activator of mTORC that we gave to the cells for 30 minutes and 1 hour. This is exokinase 2, so one of the major glycolytic enzymes that is severely downregulated in the DMXL2 knockout cells. But if we reactivate mTORC, even after 1 hour, we can already see a strong recovery of the glycolytic enzyme levels in these cells. So overall, this is a protein that seems to be at the nexus of the cellular decisions between anabolism and catabolism, and linking those decisions with the VATPase. When this protein is not there, both AMPK and mTORC signaling are perturbed, which really puts it at the core of these of both of these pathways, because it seems to be important for both and not just for one of them. And if we are able to restore mTORC activity or to restore glycolytic enzymes, or otherwise to block the acidification of these lysosomes in the absence of this protein, that seems to be beneficial, at least in cellular models. We will hopefully be testing these in vivo models soon. And that is the idea that perhaps by manipulating the levels of amino acids or glucose or the acidity of the lysosome or the activity of mTORC, these might be beneficial for patients. And with that, I have to again, invite you to see Meenakshi's poster tomorrow. Neuza will also be presenting the Batten disease story tomorrow. Thank our collaborators and the people that did the work, and thank you all for your attention.
Thank you. Any questions?
Great talk. I didn't know anything about this story. It's very interesting. So, about DMXL2. So you said it's involved in, well, it's thought to be involved in the assembly of the ATPase.
Yeah.
So I was wondering if you compare the effect of DMXL2 knockout with the effect of, that you can impair the assembly of the ATPase by mutations. So there are patients with carrying mutations of the ATPase causing follicular lymphoma that are impairing the assembly. And also there are drugs, inhibitors of the ATPase, but not catalytic inhibitors like chloroquine and doxorubicin, but inhibitor of the assembly, like Monensi, for instance, that they would do the same. So, and you consider comparing the effect of the knockout with these agents, the mutants and
so we did not, because we did, it's not the most elegant experiment, but we did the separation of the soluble versus membrane-bound subunits of the VATPase, like it was done in the 80s. And we did not see a difference in the assembly or in the distribution of the relative distribution of the subunits, suggesting that in the DMXL2 knockout cells, the VATPase seems to be assembling normally. And the fact that when we inhibit it, it's beneficial for the cells also suggests that it's acting normally. But we are now doing or planning to do lyso-IP in these cells to further verify that the enzyme is properly assembled. But that's a great experiment, actually. Yeah, we will give it a try. Sure.
Any further questions?
So we have this hypothesis, as I think you know, that the ATPase assembly, since it's an ATP-dependent protein pump, might drive recruitment of the mitochondria to provide ATP for the proton pump function. Do you see any difference in contact with mitochondria in your knockout cells? Do you see any reduction?
Yes, major reduction. I'm sorry, major increase. Am I right, Meenakshi? There's a major increase, yeah. Which is consistent with the glucose, cannot say glucose starvation because they are not glucose starved, but the inability to use glucose, yeah. So we see a major increase, actually, yeah. But maybe these cells respond in a funny way because they have all the necessary components, but they don't know that they have them. So when we manipulate, when we take glucose but leave it away, or when we take amino acids but leave them away, they don't seem to notice the difference in a canonical manner. So perhaps the massive increase in the microlyso contact is an attempt to correct something, but it's not working. We don't know that part yet, but there's a lot more.
Any further questions?
Are there ways to do exactly the opposite what you're trying to do, to stimulate DMXL2 fusion of or assembly of the VAPases, and would that stimulate lysosomal functioning?
Well, there are ways, it's been shown in yeasts that if you manipulate glucose levels, that will also change the way that will stimulate the VATPase assembly in some ways. There's chaperones that stimulate the VATPase assembly, but again, we don't see it disassembled in this case. So one thing we may try eventually is to make the situation even worse by taking DMXL1, because perhaps DMXL1 is ensuring that the VATPase is assembling, is redundant in that sense, but not able to deal with the rest of the function. So something that could be tested would be to increase the activity of chaperones that have been described to promote the assembly of the VATPase, and in that sense, see if it would help. My guess is that in this case it wouldn't, because they seem to benefit from lower activity of the enzyme.
Right, fantastic. Thank you so much.
Thank you very much. Thank you.
Lunch next door back at 2 o'clock. We thank the